2019 is the director of the Department of Medical Physics and Engineering at the Istituto Neurologico Mediterraneo, NEUROMED, RCCS. Since 2015, he is a full professor of the School of Life Science and Technology at the Huazhong University of Science and Technology in Wuhan. And since last year, he is the scientific representative of the digital imaging multimodal platform that is now an Euro-BioImaging node at NEUROMED. His research focuses on novel, portable, and dynamic digital positron emission tomography technologies, ranging from basic physically driven technological ideas, system development, and their application to preclinical, clinical, and plant imaging. And I think now time for Nicola to start with his talk.
So thank you very much for the introduction today. And the talk today will be, the target of the talk today will be mainly about positron emission tomography. And in particular, I'm going to speak to you about the opportunities in 3D digital positron emission tomography imaging. The opportunities will be from the perspective of a physicist like me, but trying to meet the requirements of the biological, clinical, and agronomical community. So that's the challenge today, which I tried to answer to. In particular, following, I think, the main ideas and philosophy of these nice cross-disciplinary interactions we are having within Euro-BioImaging. In particular, the talk is divided into 4 blocks. The first block is sort of basic, and I needed to put also because I hope and I expect there are students from different fields. So I will answer to very basic questions like what is functional imaging, what is positron emission tomography. The answer, I mean, don't expect a full lecture about that, yeah? So I will only introduce these few concepts I will need later to introduce the 3 big blocks, following blocks of the webinar, which are preclinical PET imaging for 3D modeling and verification, clinical PET imaging, 3D modeling and early diagnosis, and finally, plant PET imaging, which I think is the experimental table of dynamic PET. So if you're ready to make this short trip with me, then let us start. So the first block, what is functional imaging? What is positron emission tomography? And I start to address this question with what I think is my dream technology and my dream imaging application. So the ideal setup, or my ideal setup, of dynamic functional imaging is represented in this figure. So do you see my pointer while I speak?
Yes.
Yes. So here on the y-axis is the space, and on the x-axis I have the time. So the space it represents the distance and the size and distance between single cells, and the time represents some messages sent by the cells, some signals. Imagine if we have a very nice technology which can tackle a metabolic process within the cell, and exactly when this metabolic process is happening, when this metabolic functional process is happening, we get a very clear spike. And each cell will send us a sequence of such spikes. And from each single cell, we are going to get a sequence of spikes in time. We can make some very nice digital signal processing and extract quantitative parameters. This is, so to say, maybe the theoretical and the best ideal functional imaging technology. So it studies the time dynamics of functional processes in space-distributed cells. on the requirements on the biological perspective are quite strong. We would like to have in vivo imaging. In vivo means that all that should be done while we are alive, while these organs or these tissues or these cells are really alive, not in vitro, in vivo. Scalable from small animal, but also to clinical use, so the full subject. Spatial resolution, single cells or group of cells, we are tens of micrometers, yeah? Spatial extension, entire organ or even a full body. Time resolution, the typical timing of cellular processes. Now my friends, the biologists, will kill me, but I tell you, tens of microseconds. So you may understand this is a quite large requirement, and is not only optimistic, but is very idealistic. However, this would be a wonderful technology, and this gives an idea on our target. With positron emission tomography, we try to approach this problem, and we try to make a compromise between our aims and what we can really get. So positron emission tomography is a technique to image functional processes in vivo. So what we have are compounds which are bound to positron emitting isotopes. When these isotopes emit positrons, positron annihilates with electrons within the body and emit two clear gamma rays with a clear energy, 511 keV. These isotopes bound to specific molecules bind into the places where we are trying to investigate, and the emission of these gamma rays is detected with a ring of detectors all around. Tomographic techniques are used to reconstruct an image of the space-time distribution. Now, I make a clear example to you. Examples: Alzheimer's disease. In Alzheimer's disease, an Alzheimer's brain is characterized by the deposit of amyloids, plaques, and tau. So here on the left, you see a very normal brain. Those are neurons. And here on the right, you see amyloid plaques outside the neurons, and inside tau, neurofibrillary tangles inside the neurons. And the question is, can we reveal them in vivo? So can we reveal them on a living subject? Yes. So with PET you could. We have a specific tracer for that. There is a molecule, fluorobeta-Ben. Fluorobeta-Ben is linked with fluorine-18, which is a radioactive isotope. Fluorobeta-Ben binds to the specific amyloid parts within the cells. And so what you can get out of it is an image of the distribution of the tracer in the brain. The tracer binds with amyloid. Now, if you study for each specific region in the brain, you can study the uptake of this tracer. So you can study in time how this uptake is changing. A time signal is then produced. However, although it may look like our initial promise, you see already its averages across many cells composing a tissue. So this example then may bring me back to the requirements we had, a biological perspective. So in vivo imaging, yes. Scalable from animal models to clinical use? Of course, yes. So we have tracers for that. Spatial resolution? Yeah. Single cells or a group of cells? No. So with positron emission tomography, you may think to be limited to 0.7 millimeter to 1 millimeter, depending on the mean free path of the positron and on the main characteristics of the scanner. Spatial extension? Yes. Entire organ or full body. And then time resolution, which is today one of the key points. Can we get typical timing of cellular processes? See next slide. So well, let's zoom in with the kind of measurement we can do. I told that with positron emission tomography, we can check the tracer perfusion or binding to a specific molecular functional mechanism, and we can look at it in time. And you see here that, yeah, we can produce a time signal out of that, which we will call time activity curve. It is how the activity in the specific region we are looking at is changing with time. Now, the typical uptake of tracer, just because the metabolic processes needs to start up, is of few tens of minutes. If you look at the scale, yeah, so that is. It is averaged in space, so you cannot have the clear evidence of a single cell of a very small. So the average in space, it's around at least 1-2 millimeters. So the PET signal, but what we learn from it is that we would like to get such a time signal. The PET system needs to have a very high sensitivity in order to sample the time activity curve very, very finely in time. And in particular, to measure the first part of it, where maybe the uptake is there, but very, very small. So in this moment, the highly sensitive and fast PET technology seems to be very needed for a precise measurement of the space-time tracer. Now, why it is so important? And why today we try to focus on this part, on the time activity curve, because the way such a tracer is the way the functional dependence on how the tracer is changing with time may be very indicative of on how that specific molecular mechanism and functional mechanism is taking place in that specific part. And how do we get such a technology? In fact, in our group, we have a very specific technology, which is the digital PET technology, developed and now in our university. The digital PET technology, what is being proposed and is currently one of the main solutions to have a very, very, very fast readout from systems. The digital PET technology, which I introduced today just at a glance, it provides on-the-fly signal digitalization. The usual time signal coming out from a PET system has this shape. We have a very fast digitalization. This fast digitalization removes the typical dead time caused by analog coincidence electronics and increases the count rate performance and sensitivity of the system. So what we came up, thanks to that very specific digital technology, to a modular technology for the fast readout of PET signals. And this happens to be then a completely new technological concept for positron emission tomography. In fact, the idea is to treat positron emission tomography as a dynamic, artificial intelligence-based test bench for risk calculation based on biological processes. So we have a modular technology which is highly sensitive and with a quite good timing accuracy. We collect samples from that, and we collect data from that, and we use artificial intelligence algorithms in order to reconstruct the images and to combine it with patient information, other images, in order to get, together with imaging, a proper risk calculation. So at the end, maybe, this picture is may look like a little bit far away from the dream of having a very nice functional imaging technology, but it's really clinically oriented in order to proceed towards a very fine risk calculation. So This was an introduction in order to put, in particular, the students and people not really familiar with PET, at least on a way to follow what comes next. So, and if I would conclude this this introduction with a clear sentence, I would say, digital dynamic PET technology measures time activity curves and retrieves quantitative information of functional processes in tissues. And now we go to the next. For clinical PET imaging, 3D modeling, and verification. So we now get maybe the first example, or clear example, on how such a digital technology could bring hints into functional mechanisms. So for preclinical PET imaging, then we base the next assumptions on digital micro-PET CT, which is in Drycan. It is it's a high resolution and sensitivity device with available tracers there, FDG, fluorethylcholine, fluorocyclovine, F-DOPA, flutemetamol, fluorbetabine, fluorbetapyryl. We have dedicated image reconstruction algorithms there and textural analysis image processing service. And in particular also dedicated plant imaging modeling and techniques. Let us see what can be done using such a device. I will proceed with an example which is maybe indicating better all that. The question is, can the information of FDG F18 be correlated with proliferation in non-small cell lung cancer? In this first example, we are going to understand what are the potential with imaging so that in order to address this point. Cell proliferation rate is an important parameter for the non-small cell lung cancer diagnosis and therapy. It is in fact correlated with metastasis. Now, the key point is that usually we do biopsy. So in biopsy, we use Ki-67 for the determination of the cell proliferation rate. Biopsy is very invasive. And however, the samples, you need to sample the cancer in a very small part. So heterogeneous cancer tissues may not be really well sampled. The assumption is here the following. Can we try to make a model so that we use the PET image in order to provide information which is very similar to the biopsy one? In other terms, can PET imaging in the future substitute biopsy or help to a personalized biopsy? So in this case, if we think about FDG is the tracer for glucose consumption. Glucose consumption is not correlated in general with proliferation. So small animal imaging can be in this case used as a test bench to verify this hypothesis. Let's see how. It is possible to make the so-called xenograft models. And here we have a xenograft model with the cancer cell line A549. What is that? You take a cell which comes from a human case, and you xenograft it in the flank of the mice. Of course, although the surrounding is different and you don't have so the immune defense of the mice is also different. So T cells are missing there. However, the cancer will grow emulating a little bit what will happen in the human case. So what we did was first FDG PET scan in vivo. And you have it here. The cancer, this part is here. After 30 minutes, the scan, we extract the cancer and we repeat PET imaging. So the cancer is extracted, and right after it, PET imaging is repeated. Now you may think, is PET imaging an in vivo? Is of course. So when you remove the cancer is not living anymore. But all the metabolism, the glucose metabolism which happened until the moment when you remove the cancer is still there. So you can have a sort of perfect image of the cancer. So after removing it, after the PET, we make Ki67 staining of the cancer. Ki67 is a marker for proliferation. Let's look better what you can do. So as we told, PET imaging is a three-dimensional, it's 3D. So what you can do in vivo is to reconstruct 3D the cancer. Now, if you compare it with the image of the cancer I've shown you, yeah, you have this sort of beam, yeah? It is possible to segment it out and have a 3D. If you see here, I also opened up the image so that you can look also a little bit what is inside the cancer. In this moment, it is like from the image, you take the cancer and you can rotate and check what is inside. The ex vivo, why we needed it? We needed it because, when we develop a model, we would like to be sure that even if we develop a segmentation, that segmentation is correct, is correctly following the expectation of the truth. So you would like to have a truth. And then here, there is the immunohistological, immunohistochemical analysis. We obtained hematoxylin, so all cells distribution, and Ki67, so highly proliferating cells. In that case, and then we would like to compare. So the interesting part is that through interpolation, we could provide a three-dimensional profile of the cancers. The question is if the in vivo part, the in vivo image, first of all, is well controlled by the ex vivo one, and if it is sort of correlated with the immunohistochemical sampling, staining. How you can analyze all that? There are several techniques. The ones which is mainly working with that is textural feature extractions. So you can extract three-dimensional textural features, analyze them with principal component analysis for data reduction. It is what you see here. So all the features can be clusterized. And then you find out the clusters. So you group all the features extracted from these images and correlate only those, the main clusters of the image. The key finding which you can find is that is mainly reported in this picture here. A group of parameters doesn't show really any correlation at all between immunohistochemical and PET in vivo, and this here. Although there is a very strong correlation between ex vivo and in vivo. So this strong correlation shows that the segmentation you're doing is really correct. So you're really getting the correct cancer. Only that that set of variable has no biological importance at all. Then there is a second group of parameters, which is strongly correlated both to all cells, all cancer cells, and to the proliferating cancer cells. So those sets of parameters are usually just due to the density of the cells. They don't teach you anything about the cancer. So they just tell you about the spatial distribution of it. And finally, you can extract another small parts of parameters. Those are strongly correlating only significantly with the proliferation. You see, the correlation is strong, although more difficult to extract from the data. And this part of the parameters has a very, very small strength. But however, it's there. This is the part of the PET image which is strongly correlated with the proliferation, and which can be then important in order to help the diagnostic path. So this gives you a hint on how quantitative can be positron emission tomography. And we went through again to, we started to think about a clinical application of all that. Of course, when you speak about cancer, diagnostic is maybe important, but maybe more important is therapy. So in that case, can we apply a non-small a therapy to that cancer? So we studied the effect of cisplatin-based drugs, and the question was if PET imaging could reflect the effects of the treatment, and which is the biological mechanism at microscopic level, which brings to the fact that we can observe some specific feature in PET imaging, which is telling us if we are curing the cancer well or not. For medical doctors, sometimes residues are important. For others, it's important to see not only if the cancer is diminishing of size, but also if while diminishing in size, the proliferation inside is really died, or it is just an effect and then it will grow up again. So what we did, we repeated the same kind of experiment, but in time. And so here you see how to add the time in PET brings to much more information. So this is the evolution of the PET imaging in time for a cancer model, and this is the cancer extracted for a control group where we have no treatment at all and treated group. Of course, the first obvious thing, the treated group has a bit smaller volumes. Yeah, but that is so trivial. So if you go more in detail, the main difference between control and treated groups is not anymore in the proliferation. This is proliferation tendency, proliferation. It is a bit more in the angiogenesis. So you have a bit less, you see there are a bit less new angiogenesis, new vessels in the cancer, but apoptosis has a strong difference. So cells, if you treat them, start to die. So, and can we see that in the PET image? Yes, you apply a similar point, and you apply then the textural feature, the coarseness, and you will observe that the coarseness has a dependence on the mean vessel density. So in particular, the coarseness has a correlation with the mean vessel density, and the coarseness you will find also correlated with apoptosis. So it means that you can extract quantitative parameters from the PET image, which can be potentially used for clinical estimation. You can go even more in detail and use the full power of dynamic PET imaging. In that case, the interest is how the tracer, the cancer is uptaking the tracer with time. And you may do new modeling for that. That's an example on how that specific cancer is uptaking tracer with time. You can apply specific modeling and extract quantitative parameters from TAC modeling. In that modeling, you can find out the exchange between the different compartments and correlate the exchange rates with the proliferation indexes. It is very interesting to note that the proliferation indexes will correlate with specific parameters extracted from a specific modeling of the time activity curve. So PET may become quantitative. And what is important is that those quantities needs to be correlated or needs to have a meaning which goes well beyond the tracer uptake. You need to what we try to study, the evidence we try to find is that those parameters have a very strong biological meaning, which is mainly related to the main characteristics of the functional processes in that tissue. So, if I need to conclude this second part, I would say that preclinical PET is a test bench for the connection between microscopic imaging at cellular level and functional imaging exportable to in vivo clinical use. So this is how we try to use preclinical PET. When we try to go to the clinical imaging is more delicate. Clinical imaging then is a 3D modeling for the serious case of early diagnostics. So it cannot be anymore a test bench. In clinical imaging, we apply all that. I focus today, for example, to the brain PET. That's a digital brain PET we realized with the same digital technology I was showing you in the introduction. It is a high sensitivity brain PET with digital technology. It has a spatial resolution, which in this specific prototype is approximately 1.5, 1.6 millimeters. That's limited by the crystal size, maybe in this version, mainly in this version of the system. It's not a limitation of the technology at all. It can be done in continuous trigger mode. So dynamic adjustment of coincidence time and energy window in offline analysis. This brings the sensitivity to be at a quite high and competitive level. And this is all what we need for good dynamic imaging of the brain. Let's start first. Let's start again with an example. Our first example here is a study we did where we asked to ourselves if, or better, if the clinician asked to us if we could find any hint on how to correlate the uptake of fluorbetabine with the age of onset of Alzheimer. Fluorbetabine is the tracer which we have shown in the introduction. It is linking to amyloid. So the question is, on the clinical point of view, early onset Alzheimer's disease patients have Alzheimer's disease progression, which is much more aggressive than late onset Alzheimer's disease patients. So there is no obvious difference usually between early onset Alzheimer's disease and late onset Alzheimer's disease, even using two tracers. So here you can see the example. This is one part of the brain, the lateral orbital gyrus. The upper row is the early onset Alzheimer's disease, and the lower row is the late onset Alzheimer's disease. Could you see something? Actually, I cannot. So if you just look to the image as it is, that's the amyloid uptake. So the fluorbetabine uptake. So it should reflect the amyloid distribution in some sense. The two sets have no difference at all. And the question is the usual, can we make a dedicated analysis to the image? So the analysis we provided then is the following. We can extract textural features from each brain region. So we segment, there are some standard templates for that. We can segment, get one of the brain regions, extract textural features out of that. Reduce the data because in that case we have almost 9,000 parameters. Reduce them with principal component analysis and look for a set of parameters which are sort of correlating with age. You may understand that there are some parts of the brain where, in fact, amyloid shows a very clear correlation with age. And not the uptake of amyloid, but the way amyloid is in space distributed. So, in particular, we found out that, so here in blue, you have the Alzheimer's disease patients, in black, the background, so the control group of patients without Alzheimer's. And we found out that a strong age correlation is evident for some groups of specific markers from the PET image extracted with textural feature in the anterior temporal lobe, superior temporal gyrus, in the frontal lobes, which are characteristics of all parts, which are also very characteristics of Alzheimer's disease. And so such information can be, and such specific quantitative information out from the PET image can be used then to develop a clinical digital imaging biomarker. So here, PET system, the PET image information can be then bridged to all the other clinical information. Because what we did until now is to take the image and to take out parameters out of it. The parameters are numbers and which characterize that image and that patient. But together with that number, we may have much more information characterizing the clinical state of the patient. For instance, here is a study which we started within the Gatekeeper project, EU project, biomarkers for early diagnosis of Parkinson's disease, where positron emission tomography information can be used within the clinical trial, the diagnostic path. So the problem was there that at subclinical stage, Parkinson patients are affected by non-specific symptoms such as sleep disruption. So is it possible to identify biomarkers for the early diagnosis of Parkinson's disease? So a patient has an anamnesis first, will go to the medical doctor and tells his initial non-specific symptoms, sleep disorders, depression, mainly. We can make blood analysis. We developed a specific blood metabolomic analysis tool, which from 630 parameter may give you a sort of Parkinson's disease risk indication. But if you have such a risk, then we have specific tracers, dopamine tracers, which are binding exactly to the problem which are causing Parkinson's disease. So you can add at a very early stage PET imaging. And in that case, you may have a very clear risk assessment. That's the idea. Now, how we do that? We extract textual features for the relevance part of fluorodopam. Here you have an example of where F-DOPA is specifically the specific uptake of F-DOPA in the brain, and we combine those textual features with blood biomarkers to make a full model. We found that, in fact, not only we could extract such features and we could combine, but we could use machine learning methods, supervised but not so complicated, like OPLS, in order to distinguish quite clearly early Parkinson's disease subject from healthy groups. Here you can see how well the 2 points are divided. And for to be intellectually honest, I also put I also added the statistical evidence that the model is not overfitting on the right, while the model on the left may be overfitting, on the right not. So it is possible then to strongly distinguish between. And we can add even more information there. We can ask to ourselves if, thanks to the metabolomic biomarkers, we could have a specific metabolic pathway which is strongly related with such patients. And so what we did, we got the important metabolomic parameters, and we correlated them with age. And we found out, you see here in the last picture, that in particular, glycine, threonine, and serine metabolism correlates with age, all in Parkinson patients. After we got that, we went to our friends, to our neurologists, who gave us all this fantastic data set. And we communicated with them that it seemed so. And in fact, when you look to all the story from a clinical and medical perspective, glycine, threonine, and serine metabolism are strongly involved at the beginning of Parkinson's disease. The interesting point is that we find out everything and merge it with PET imaging, which in fact apparently has sort of nothing to do directly with all this. So how to how to go forward. So the point of this talk, of this seminar today, is also to show a bit more perspective. So how can we have then a technology which is sort of truly dynamic for brain? And here is the future, or what is happening right now, a completely new device. A device which is a helmet, a PET imaging, a PET system, which is helmet shaped, which can follow the patient. It can go up and down. The semisphere scanner fits the human head perfectly. The height can be adjusted, so the patient can sit, but can also stand, can move freely while taking a PET signal, while taking a PET image. Multi-angle imaging can be also achieved by rotating the gantry, and the device is fully portable and can also follow the patient. In this case, we can have not only dynamic PET imaging, but a dynamic PET imaging while the patient, the patient is is freely moving or doing a specific action, which is mainly which may be linked to that specific disease. And out of that study, how that action could modify the specific uptake in that specific moment. It is not a dream. I show you now the first clinical trials we are doing in Wuhan. So you hear also the noise from the PET room. That's the first. That's patient 0. So the first subject, which is going through to the scan, and you see she's freely reading, for example, her book while taking the scan. That I believe is the future of dynamic PET imaging for the brain. And studies and data taking is now still going on in this frontier imaging technique. So if I need to conclude this third part, I would say that the clinical brain PET brings dynamic functional imaging to the clinics in order to explore early diagnostic paths. That is the mission. And this is what I try to show you through a few examples. And now we go to the last part for this last, I think, 6 minutes, which I'm left. So plant PET imaging. Plant PET is the experimental test bench for dynamic PET. You can ask to yourself why to do a PET to a plant. So I had the same question when my good colleague of mine and friend, an agronomist, asked me if it was possible to make a PET to a plant. So in fact, it became a new frontier of dynamic functional techniques. So the key problem is that biotic and abiotic stress reduces the yield of plants with consequent reduction of the production. And the key question is if we can visualize the effect of this stress very early before any symptoms occur. So again, early diagnostic. Yeah, good. From qualitative dynamic imaging, many techniques can be used. In fact, so if you look, I said we summarize it in this picture. The many imaging techniques which are already used in agronomy to check this problem. Qualitative, on drones, they measure reflectance, to quantitative, metabolomics. Yeah, metabolomics is used also very normally in agronomy. So to study but metabolomics is disruptive. CT and MRI, there are examples. So it's possible to have structural imaging from the structure of plants, maybe understand if something is going on. But again, the only in vivo dynamic technique for that is PET. So, PET is looking at a very specific thing in a plant. So here is an example. This is a normal plant, and this is a stressed plant. The plant was stressed with salt. So there was lots of salt in the water. So if you look, visual expression, they look very similar. Even as non-agronomist, if I look to the roots here, they look even better, or the stressed plant look even better than the normal one. So the visual inspection is still the one which is strongly used for that. Yeah, so in agronomy. Then we have morphological information. You can make a CT. This is a CT scan exactly of those two plants. We did it with a micro CT in the lab. So you see here in green is where the water flows. So the plant vessels are O-shaped in the stem. So, and again, morphologic information will not show you exactly that there is any difference. The problem is that it's very early to find a difference. It's only 10 days. It's only 10 days. But when we get to the functional quantitative PET, so when we look how and the water is flowing through the plant at a quite good resolution, then it is possible to see quite immediately a strong difference between the normal plant here on the left and the stressed plant here on the right. And this is only looking how the tracer is flowing through the plant. So this is the simplest example of plant PET imaging, where we have a tracer flowing to the plant vessel. How we do that? So how it works, you have a plant, tracer is transported, diffused, and locally stored in the plant. So while during this movement, we have a plant PET around, so we measure the activity. Of course, it should be dynamic. It's 3D. It's 3D. At the end, you have a very nice 3D image of the plant, but you cannot do anything just looking at a nice 3D image of the plant. The key information is on how, in time, this image is changing. So we extract from a small part of it. We extract region of interests, and from each region of interest, we get a time activity curve. After that, we developed kinetic modeling, and we can quantitatively calculate the kinetics of the tracer through the vessels. What we reconstruct everything. We reconstruct the tracer profile, both the tracer flowing here in red and the tracer which is stored or which is diffused. And we reconstruct the velocity profile, which is in particular, in this particular example, is flat, and this is quite small. Oh, sorry, here is velocity. The next step will be to combine with the immunohistochemistry in order to give biological grounds to all the findings which we get from quantitative time dynamic PET. It is a challenging technology. So plant PET needs to face lots of many differences between preclinical and clinical imaging. Plants don't have a very good immediate self-adaptation system for when the environment becomes very cold. So there is a strong experimental limitation. Light, temperature, humidity affect the measurement a lot. So tracer decays much faster than the displacement processes sometimes. And then, of course, there are several technological limitations related to the design. What we have is a completely transportable plant PET. This is my hand, and it stays, yeah, if you look at the sides, yeah, it stays in my hand. This is how we start to make now taking the first measurement of it in order to have a system which is truly detecting the kinetics of tracers inside plant for a quantitative approach in precision digital agriculture. So, to conclude also this last part, plant PET is the perfect test bench for kinetic modeling of dynamic PET signals. And with this, then, I can only tell you that I invite anyone who likes to join our research and also to use all the technologies I've shown to you in our node in NEUROMED, where we have animal facility, animal facility in the science park, technology computing, the population imaging part, training, and here you have an aerial view of the entire campus. And I thank you for all. Here are our contacts. Thank you very much. And I think now we can open the floor to questions.